So I welcome you to my seventh lecture. I mean, seventh lecture that's in bio and organic chemistry. So an important aspect of bio and organic chemistry is use these metals in medicine. I told you how this some of the proteins, whatever proteins that exist in nature, can act as anti-cancer agents, right? So the knowledge that we gained through metal DNA interaction, you know, has been used to devise metal-based anti-cancer agents. So in our laboratory, took up, we made a large number of copper and ruthenium complexes, and we thought they can act as anti-cancer agents. So in order to see whether they can act as anti-cancer agents or not, we investigated their metal DNA interaction. That means, very crudely I can say, if a metal complex interacts with DNA very strongly, there is every possibility that the complex can act as an anti-cancer agent. This is what we verified in our laboratory. So we devised a large number of copper compounds, ruthenium compounds. From the experience we had, the principles we understood from metal DNA interaction. So this cancer, you know very well, is a very dreadful disease, right? According to WHO's report, World Health Organization report in 2012, so this I prepared a very long back. So cancer is the leading cause of death worldwide, accounting for 9.6 million deaths. Okay, in 2012. We also learned that deaths from cancer worldwide are projected to continue rising, with an estimated 13.1 million deaths in 2030. In India also, such a type of account has been given. Probably within this week, there was news in Hindu newspaper that the death rate is also increasing in India, right? So these are the types of cancers. For men, women are affected by different types of cancer. The most important type of cancer in women is lung and breast cancer, right? In men, you have lung and bronchus cancer, and there are also all other cancers, kidney and other things. All cancers are there. So, a lot of data is available on this particular aspect. I tried to get the latest figures, but I couldn't get them from Google. I tried Google up. But anyway, this is rising. Still, the rate is increasing, right? So cancer is one of the most common diseases in the developed world, right? 1 in 4 deaths is due to cancer. So 1 in 17 deaths are due to lung cancer. Now you know very well that it's COVID-19 that also brings about attacks the lungs, right? So there is a very vulnerable organ of human beings, right? So lung cancer is the most common one in men. Breast cancer is the most common one in women, right? So we have used, in our studies, cancer cells from these two diseases, right? Cancer cells, and we have used them in our experiment. That became possible because of an interdisciplinary work. We had Professor Akbar Shah in biology, and we interacted with him, with these people in the laboratory. So we were able to interact very strongly for a large number of copper-2 compounds, lithium compounds we made, and we had published our papers, more than 30 papers also we have published them. Even Professor Akbar Shah is also contacted very well by others, so he has also published, apart from ours, a number of papers. So This type of work is an interdisciplinary work that became possible only through the interaction of students. My own student, Professor Rajendran, who is now Central University of Tiruvarur, Tamil Nadu, he was able to interact with his friend, Professor Akbar sir. That is how this interaction happened, and we had a very fruitful interaction, and we had a very useful molecules to be suggested as anti-cancer agent. So youngsters here should know what is meant by cancer. So cancer is uncontrolled cell growth. When we have normal cell division, right? No problem, right? See, we have cell growth happening like this. But when you have a cell damage, again due to the DNA, right? There's a defect in DNA. The cell undergoes damage, and the cell commits suicide. The cells will commit suicide or undergo apoptosis. So this is called DNA repairing. So whenever there is a problem, it will be repaired. Okay. In fact, I'll just come back. So this type of repairing, the mechanisms are known, right? When this type of repair fails, okay, this cancer gel, this one will start dividing like this. Okay, they will start dividing like this. So in that case, you have uncontrolled growth. Okay, so abnormal cells like this divide without control and are able to invade other tissues. So this is normal cell division. When it is abnormal, there will be some triggering molecules which will ask this cell to commit suicide. That is called apoptosis. So because of this mechanism, we are able, people like us, we don't have cancer. We also generate this type of damaged cells, but they are repaired. Cisplatin is the drug which repairs such cells. Okay, so now you know what is cancer, uncontrolled cell growth. See, so what is the origin of this cancer cell? Cancer. So where we should target, what we should take, what is the theory we should use to, if at all you want to, devise new molecules, new as anti-cancer agents. So the answer lies in the DNA. So whenever there is a defect, it leads to, uncontrolled division of cells and then tissues, right? Will be grown. So a number of metallo drugs are known for cancer. It's an accidental discovery. Cisplatin, is the best reagent, still known worldwide, readily available in the market as an anti-cancer drug. Is good for testicular, bladder, lung, gullet, stomach, and ovarian cancer. Okay, and people have, in fact, a large number of patent compounds have been made to displace this particular, to substitute this cisplatin, because there are side effects also. There are severe side effects of this drug. Okay, so the urinal problem will be there, right? When the excess cisplatin is excreted, you will have problem. So people have tried to, substitute this with carboplatin, second generation, and oxaliplatin, third generation. So in spite of these drugs, cisplatin is used. I mean, it is sold very extensively in the US, cisplatin. Sometimes this cisplatin is mixed with Taxol, the organic compound, natural organic compound, which is having a very wide application for all types of cancer. So this cisplatin can be mixed with Taxol and it can be used as a drug. So we just visit some of the hospitals in Trichy itself. You can see there they have written what are the chemotherapeutic reagents, chemotherapeutic drugs they use, how do they mix them and all. So these are the drugs which are widely sold, having a big market in the US and other countries. So how does this cisplatin act? As I told you in my previous lecture, it is chemists like Stephen J. Lippard in MIT who worked on this type of molecules and brought about a theory on the biologic basis of chemotherapy. Now this has become a textbook aspect for MSc students. So what is done? This cisplatin is neutral, right? That passively, passive diffusion, they go into the cell. This is the cell membrane, passes into the cell because it is hydrophobic also because its charge is nil, no, it's zero charge. It goes into the, and this undergoes hydrolysis. So you have a replace simple substitution reaction which you have learned in inorganic chemistry. So water, right? So this one has two displaceable ligands, right? So they can be used to bind to the DNA, which is a wrong DNA, repaired DNA, which has defects, right? So when this compound, we can also use another one to redisplace two by solvent. They just this one interacts with DNA very fast. So this cisplatin can interact with this defective DNA very strongly. See, the DNA is kinked. So after this, it repairs the DNA. How it is repaired? Actually, it's involving a lot of mechanisms, right? So the DNA is repaired, right? So when it is unable to repair this DNA, it is coming out of this component, and then it is eaten up by Golgi apparatus, right? So the cisplatin, functions by repairing the DNA. So platinum will be excreted. That is where problems are there. So people want to replace platinum by other compounds, metal drugs containing copper or ruthenium. Ruthenium is inert compound, so it can easily pass through the cell like this and take part in such repair. Copper compounds, copper is there already in our body. It's bioessence, biocompatible. So people try to make copper compounds. We also made a lot of copper compounds. Try to substitute this cisplatin, right? So these are the people who received the Nobel Prize in 2015 for their work on this type of repairing, not by cisplatin, by overall mechanism. So they were recognized for this Nobel Prize in 2015 for mechanistic studies of DNA repair. So a lot of things are known. Umpteen number of proteins are involved. They are activated within the cell, right? It has become a bioinformatics subject. So a lot of proteins inside, they are all activated. We have used these proteins also in our studies to say that how our compounds activate a rain of proteins, creating a rain of proteins. So this is what we have deeply investigated, but for your purpose, I shall only point out our results. So chemotherapeutic agents is medicinal chemistry. So a very good drug like cisplatin should be able to induce efficient apoptosis in cancer. It should make the cancer cell to commit suicide. So all our compounds, copper compounds, like cisplatin, we have found they bring about apoptosis. Okay? So apoptosis is the process by which cells maintain a balance between proliferation and death, often induced by external and internal stimuli. Okay? There are stimuli, right? So these chemotherapeutic agents, as I said here, they should be able to bind to DNA. That's one prerequisite. That's what is the theory, right? So they should be able to interact with the DNA and proteins also. That's what we have done. We have taken our compounds and interacted them with the DNA. Whenever we find that the compounds, copper compounds, or ruthenium compounds bind strongly to DNA, we have investigated their ability to damage DNA. Whether they are able to cleave DNA very strongly, right? Again, I told you in my previous lecture that they should be able to bring in, double nick, double cut in the DNA. In that case, it's a better reagent, better reagent, I mean, a medicinal reagent, I mean, the drug. So they should be able to damage DNA irreversibly. So we have tried to make compounds and try to explore the relationship between DNA cleavage and damage potential. Right? Different copper compounds, depending on the ligand system, they have different ability to cleave DNA. So biochemical activities in cancer cells, that also we have evaluated. And structural conformational changes that we have noticed for this 5,6-DMP compound, such changes should be are affected by compounds or copper compound, then they are better, anti-cancer, they function as better anti-cancer agents or cytotoxic agents. So cytotoxic agents are agents which bring about the destruction of the cell. Destruction of the DNA molecule. When it is a good cytotoxic agent, it can behave as a good chemotherapeutic or anti-cancer agent. So cytotoxicity is different, anti-cancer activity is different. First, we should show that the compounds are cytotoxic, toxic to cells, toxic to DNA. Then we can go and test whether they can act as anti-cancer agent. So if my compound is good, we have to wait for 50 years to see whether it can be used as an anti-cancer. So a large number of tests, clinical trials, as we see, the COVID-19 also, our drugs are undergoing phase 2 and phase 3 trials nowadays. Right? So those trials are to be done, and we have to establish that they have a very lower amount of side effects, lower side effects. That we have to prove. Then it will be taken to the treatment stage. So what is that we did? We made a large number of copper compounds. All of them contain phenanthroline. In my previous lecture, I showed you this ligand system. It's a very good moiety of the complex which can try to bind to DNA, recognize and interact non-covalently by inserting itself in between the base pairs. So it is a good recognition element for the compound. And we have also shown that this can bring about conformational change when you have dimethyl group. The copper can be 2 plus or 0. See, this can be 0. If this is 0, it can easily pass through the biomembrane, cell membrane, and reach the cytoplasm. So we have to look into all these possibilities, right? So we devised a mixed ligand compound. One ligand is phenanthroline, the other is an amino acid, for example, NHTC overhead. An amino acid can be there because amino acid is there present in us. So the various components here are mostly, biocompatible, except this phenanthroline. We have designed several ligands which will make these molecules water soluble, anchor the metal ion, right? And this may also interfere with DNA binding, right? So we have isolated ruthenium compounds, copper, iron, cobalt, nickel. We have studied their ability to DNA, bring about DNA damage. They also play a role, not only these ligands. So most of the compounds we have shown, they bring about apoptotic acid. So we have published papers from our Bharathasan University in very leading journals. So we are all we are quoted for our theories and other aspects we have established in our library from these studies. So we have published 2 papers in 2020 and also 2019 also. 2 or 4 papers we have published, right? Even now we have published papers. So we have become pioneers in India to show that copper compounds can act as anti-cancer agents. I shall take up some typical example and then illustrate the methodology we make. So this is copper complex, right? What is this one? You know very well. This is tyrosine amino acid. Amino acid, right? Like glycine, side chain is phenolic group. So it is a tyrosine amino acid. This is the phenanthrene and other ligands which we chose as the answer, the co-ligands, right? So we isolated a large number of complexes with this type of ligands. So this is 5-6 diameter, which I pointed out. They have a special type of interaction with the DNA. So this is the ring system, phenolic ring system. See, in the metal complex, this is placed over the phenanthrene ring system, a pi-pi interaction. So this is stabilized. So we made a large number of compounds. Phenanthrene is expected to insert itself partially into the DNA and stabilize its binding. See, we have synthesized another type of ligand. We have extended the ring structure here, the planar ring structure. So this is called DPQ ligand. So this will be able to bind much better than this. This will also be able to bind much better than this because this will use hydrophobic binding. This will make use of a stronger partial intercalation. This is what we established. All these DPQ complexes have a higher binding constant than these phenanthrene. These compounds had higher binding constant than this phenanthrene. And we were able to show that most of our compounds, these two type compounds, copper compounds with these two type of ligands, had higher binding constant, higher protein binding constant, and then they got a higher ability to cleave DNA, and their ability to bring about the destruction of DNA. Cleavage of DNA is also very good. So we were able to show it throughout our studies that these copper molecules, we can reduce this copper 2 to copper 1, that can bind much better than copper 2 form to DNA, and that copper 1 form will bring about DNA cleavage like your bisphenanthrene copper 2. So what we do, we isolate these compounds, put them in solution, study the X-ray structure of the solid state, put them in our buffer, twist buffer, as biologists do, put them in buffer, we do their electrochemistry, perform the electrochemistry, ability of copper 2 to undergo redox to copper 1. We investigate the structure in solution also using very sophisticated techniques like EPR technique, right? And we use UV visible and other techniques to investigate the way in which these molecules interact with DNA, right? First, we establish their ability to bind to DNA. We then establish whether they will be able to cleave DNA. Then we establish whether they will be able to, kill the cancers, right? Then the next thing is, fourth thing is still more sophisticated one. If they are able to bring together the cell death at very low concentration of these complexes, then we try to study at what stage of the cell growth we bring about these molecules bring about the cell damage. That is called as a function of time we brought about this. So this is of the methodology we use. I shall illustrate this methodology. So we use UV visible spectral studies, viscosity measurements, right? When you have DPQ here, this methyl here, they behave differently from phenanthrene compounds. They have a higher binding constant, right? So the DPQ compounds bind more strongly than 5,6-DMP than phenanthrene. So these ligands are able to recognize much better than phenanthrene. So we are able to publish very good papers, running to 20 pages in leading journals like Inorganic Chemistry in 2009. So our papers are quoted very well because we have used very interesting biological experiments. See, cleavage. We have used cleavage. DPQ complexes are much better DNA cleaving agents in the presence of ascorbic acid, which reduces copper 2 to copper 1. See, the one interesting thing is, without the addition of thiol or ascorbic acid, we are able to generate, these molecules were able to stabilize copper. without the reductant. See, they were able to cleave DNA. That is the most interesting thing about our compound. So when we use this as drug, we need not add a reducing agent. Again, we see DPQ complex, 5,6-DMP complexes are able to bind DNA very strongly. They bring about cleavage also very efficiently. One moment, please. Then we studied the anti-cancer activity. We mix them with lung cancer cell lines, right? And then study their ability. You can see the ability to kill cancer cells is compared here. 5,6-DMP is much better, right? So what is the amount of compound we need, the drug we need, the molecule we need to bring about death of the cancer cell? That is called IC50. What is IC50? The amount of the complex needed to kill 50% of the cancer cell. That is defined as IC50. See, our tyrosine compound, 5,6-D compound, needed only 2.4 micromolar. This nanomolar, 2.44 micromolar. That is very small concentration. Whereas cisplatin needs 60 micromolar. Whereas our compound needs only 2.4 micromolar. Specifically, all other phenolphthalein compounds need higher amounts, but these two compounds need only very small amount to bring about cell death. Within the cell death, 50% of the cells to commit suicide within 24 hours. Right? So in vitro cytotoxicity was performed for all the complexes. So 3 and 4 with special designed ligands, they are able to kill cancer cells. Right? See, we also studied at what stage the molecules bring about apoptosis. See, when we treat the cells and then follow their time course of the cell death, see these are small. This is the control cell, no apoptosis. But when we mix these cells with our complexes, you can see they undergo cell division. The smaller ones, young cells are there. That means you have apoptotic body formation. So our drug asks the molecule to commit suicide and cell division occurs. So the this AO, ethidium bromide, acrid orange staining experiments against the cells illustrate that our compounds are able to induce DNA fragmentation and apoptotic body formation. That is wonderful thing. Our cisplatin is also able to bring about this type of cell death in apoptosis, but at higher concentration. So the percentage of abnormal nuclear apoptosis higher for 5,6-DMP than for DPQ. See, 5,6-DMP, which we showed as a very specific reagent to bring in conformational changes on DNA, they turn out to be good cytotoxic agents, so they can very well be better anti-cancer agents. So this is what we saw. FACS analysis, flow cytometry analysis we found. So we found that that particular, our 5,6-DMP compound brings about mitotic catastrophe through apoptosis. Right? So this is what we saw in FACS analysis with the help of people from Bangalore. That's where we have the unit to do such study. That's biology unit for cell studies, etc. in NCBS, National Center for Biological Studies in Bangalore, near Raman Research Institute. And there we studied, there we can study this FACS analysis, flow cytometry. As a function of time, we can study the cell growth and at what stage our molecules bring about this cell. Here is another molecule which we made, ligand system. This is Professor Rajendran, who interacted with Dr. Periyasamy in Professor Akbar Sah's laboratory. We made very simple compounds like this because these compounds made copper compounds soluble, phenolate compounds. We have tyrosine, right, in our body. So amino acid. So we made these compounds, bipyridine and all, TMP, and then DPQ. We determined their structures, right? And then we investigated their ability to bind to DNA, right? This also we published in inorganic chemistry. They were able to bring about DNA cleavage. Again, see, with these compounds, with methyl groups and extended ring system, they were able to cleave DNA much better. Look at the amount of complex we needed to bring about cell group death when you have 4 methyl groups. So cytotox studies against cervical cancer, right? Then we need only 0.85 micromolar IC50 value, whereas cisplatin and mitomycin, another drug, is known to use higher amounts for treatment, right? But our molecule needs only 0.4 micromolar, very small. See, 48 hours, we need only very small amount of this. See, so this is what we saw, that our molecules are much better than already used drugs, right? More potent than cisplatin and mitomycin. So this is how the molecules bring about cytological changes, cell death, etc. This is how the molecules become big, right? How apoptotic bodies are there. So this was demonstrated by our studies. At very low concentration, 0.25 micromolar. This is a standard, uncontrolled, without treating our cells, without treated by our comp. But when you treat them, you can see apoptotic. So we are able to clearly show that these molecules bring about cleavage or apoptotic body formation. See, this is this flow cytometry. At what stage we are able to bring the cell death in here? Well, this is somewhat difficult. I need more time to illustrate this one. So these are the conclusions we made. We have also shown that these molecules, the dimethyl compound, is able to bring conformational change on DNA and cleavage. So nowadays we work on copper-2 compounds of other drugs like nalidixic acid. This nalidixic acid is here. It is a first generation quinolone antibacterial drug, right? So this is the drug, which we made as a co-ligand with phenolphthalein, right? So we combined this activity of this drug along with phenolphthalein. We are able to publish very good papers in 2017 in Journal of Inorganic Biochemistry. We have now published more papers in Inorganic Chemistry Acta and Dalton Transactions, making use of this nalidixic acid. This has already earned very good number of citations for our work. This is Loganathan, who is now is returned from Purdue University in the US, my student. He's an associate professor in VIT in Vellore. He's making a very novel study. One of his work, he was involved in one of the very important work whereby he has made targeted molecules in Purdue University, right? He was a part of the medicinal chemistry department there, and he was able to assist in making some newer molecules, which when bound to the cancer cells, they started emitting light. See, the cancer cells have some, receptors, like your COVID-19 is a virus having some strong receptors on the surface of the virus, right? Similarly, the cancer cells have good receptors. These receptors, can attract these newer drugs. So the drug, the molecule that was with which he was associated, bind to these cancer cells, and the cancer cell tries to emit light, yellow light, like this color, right? Phosphorescence. So this has an advantage. See, the doctors, oncologists, they have difficulty in removing the mass, the cancerous mass. But now, when the cancerous mass is treated with this particular drug molecule, with which developed by this Loganathan, so this molecule emits light. So doctors can now easily segregate, differentiate the normal cell and then this cancerous cell. So these cancerous cells can easily be dissected, cut and removed. So that is how he has made some contribution. He's published his paper in this discovery in Nature, some medicinal Nature journal. So he has got a DBT award, Ramananda Swami award for 5 years and money. So he has been attracted by VIT and he works there to design newer molecules in this direction. So youngsters can approach him to do a PhD with him, right? Loganathan. That's Rangasamy Loganathan. He's from PSG College. He joined me in Bharathidas University, and then he was with me in Central University of Tamil Nadu. So he has done a lot of other important work like this. So this nalidic acid complex found out to be there. These ligands themselves have anti-tumor activity. See, our goal is to make copper compounds and make it a targeted one to the cell. So copper compound can bring about the cleavage very well, right? By generating copper 1. And this one is known to stop the DNA replication. You know what is memory replication now? It stops DNA replication, this ligand, smaller nalidic cell. So our copper compounds should also be able to do that. So these are in generic name, we call them as quinolones, antibacterial quinolones. So what we have done in our laboratory now, we have varied these quinolones. Tejpur University, which I visit often as a professor of eminence, there we have a student, Mithu Sarma. She has varied all these substituents and we have made large number of copper compounds. We have shown that those molecules are even much better than nalidic acid. So these copper to quinolone complexes will enhance DNA fragmentation and apoptotic activity, cell committing suicide, triggering the cancerous cell to commit suicide. That is much better activity than compounds which may. This is the drug nalidic acid. It is used for as an antimicrobial, gastrointestinal tract and other drugs, antifungal, antiviral, anti-inflammatory. So quite a large number of quinolone ligands are known. So we are trying to take them as co-ligands along with phenanthroline. So this is the one we use. Look at that. So this nalidic acid can bind like this. And these phenanthroline bipolar compounds can bind here. We have a water molecule, right? These are the ligands. We have shown throughout our study, it is this axially bound water molecule which is able to bring about the damage of DNA. It is able to bind and do hydrolytic cleavage of DNA. See, that is how this bond is a very wonderful bond. Right? This is responsible for the cleavage. So we have a water molecule there. Right? The bis complex of this nalidic acid is insoluble in water. We have published its execution now in ICA. So that cannot act as a drug, very good drug, but this one is better. It's water soluble. It's square planar. 2 oxygen, 2 nitrogen like this. CN2, O2. We have established their structure with EPR spectroscopy in solution and in the presence of our buffers we use for all our DNA binding experiments. Let us look at the results. So this copper phenanthroline compound is able to bind to DNA. That is what it shows. We have a UV signal, UV response for the complex. When we add DNA, that signal disappears. Right? See, when you add phenanthroline complex of this nalidic acid, this maximum by 10 or 15 nanometers. This means that this phenanthroline intercalates partially with the DNA. A lot of people use this technique to characterize the binding with the DNA. This is a very important characteristic which is important to say that this phenanthralene partially intercalates. A lot of people publish papers from our own country saying that the complexes intercalate, partially intercalate, without proving that this type of shift, we call it as red shift, from UV to the near IR or IR region. So that is shifted. See, this is called red shift. This red shift is an index. It's a signature of the partial intercalation of phenanthralene. So I take some time to explain this because people who hear my lecture should be able to understand what are all the different criteria we make use of to characterize the binding. So this phenanthralene ring is able to depack, this large shift, 10 nanometer. So there's the phenanthralene stakes with the frayed DNA. Frayed means the base pairs, when they are packed like this, they come out like this. So that's called fraying. So it induces fraying of the base pairs. It's able to insert very well, and this phenanthralene is able to get into the DNA, whereas the nalidate component faces the solution. So if you take, for example, a DNA like this, the phenanthralene one here binds the DNA, the nalidic side faces the solution, right, and brings about redox everything. Whereas the molecule has stuck itself, bound itself to the phenanthralene. This type of binding also we have tried to do, like docking studies. See, all the molecules, this is the DNA. Computational, you can, bioinformatics people can do this. Computationally, you can regenerate a synthetic molecule like this for this length. So our molecules, see, they were able to be located here. See, the phenanthralene is present in between the base pairs. So the base pairs here will be frayed, they can bind. See, when you have differ the substituents, see, they have different bindings. So we are able to show that these molecules can recognize different sites on the DNA. See, these are the water molecules I showed you, bound to copper. Suppose you remove the water molecule, what happens? Then the binding capacity, binding mode changes, right? So see this is the non-aqua complex. Non-aqua complex without water. We remove the water computationally and we try to locate how they bind. Right? So we were able to establish that they are also binding strongly. So what is that you find here in this? Double strand DNA cleavage without the reducing agent. We need not add any reagent to convert copper to copper 1. Enough if you add, you need not add ascorbic acid. That's what we added. We brought about the cleavage. So you can see this is uncut DNA, plasmid DNA, uncut. When you spot it at this point, it moves like this without any cut. Because this is more hydrophobic, moves very well. Whereas this is a linear DNA, which has undergone 2 cuts. That is, can move in the electrophoresis experiment very much better. So this is NC DNA, only 1 cut, right? So there is no DNA available here, original DNA. See, that is not here. So this is a very good illustration that our compound is able to completely break down the plasmid DNA, cut the DNA. The interesting aspect is you get this linear DNA. One can very well see that this linear DNA will find it difficult to go back to this DNA, circular DNA, and then to this plasmid DNA. So irreversible cutting has come. So nalidic acid is a metal complexes are better reagents, right? They bring about cleavage even at 100 micromolar concentration, very low concentration. Our compounds is sufficient to bring about the cleavage of DNA. That's what we saw. And when you add reductant, the cleavage is much better. Which complex is much better? 5,6-DM dimethyl compound. I told you that is able to have a special recognition, site recognized on the DNA. Take this 3478 DMP, that is also able to bind much more strongly on the DNA. See, now let us look at the IC50. The amount of the complex we have to add to bring about 50% cell death. See, which compound is having a higher, very low value? It is the TMP compound, 4.2 micromolar. Cisplatin, under the same experimental conditions, it only 6.7 micromolar. So the TMP complex, which is hydrophobic, can easily pass through the biomembrane, right? And this study is consistent with our DNA binding strength. This is this complex which is having a higher DNA binding constant. So now you can conclude that this complex with a higher binding ability, DNA binding ability, can bring about, can be act as a better potent cytotoxic agent. So the TMP complex shows cytotoxic. So if you want to design a good, anti-cancer agent based on copper, then you have to choose. You can choose TMP ligand bound to copper, or 5,6-dimethyl compound bound to copper. Right? So we made further studies. Quest 33258 staining studies. See, when the cells, control cells, right? So when the cells were treated with this one, right, the cancer cells, you see, this is the control cell. No, nothing has happened. This normal cell. But when they are treated with our 3478 TMP complex, you can see apoptotic body. The cell have undergone apoptosis. Cell is committed suicide, right? That means our drug induces these cancer cells to undergo suicide. So that is the best drug. So this hydrophobicity of these particular methyl groups, polygan, facilitates the transport of the complex across the cell membrane and gets released at various organelles present within the cell. They just involve in DNA binding and then leads to apoptosis. This is some idea we have about the mechanistic details. How do they bring about mechanism? This is only a crude mechanism. Detailed one will be involving a large number of proteins available within the cell. That also we have established by sophisticated experiment. So what we have shown? We have shown that mixed ligand compounds are becoming water soluble. See that? Whereas copper compounds of malodic acid is insoluble. So these compounds with phenolphthalein, they stay well frayed AT base pairs. That's what we have shown. This compound is able to bind to minor groove and then bring about the DNA damage, right? And these compounds are much better because they can self-activate double-strand DNA. It's called self-activation. You don't need to do any reagent to get the copper 1 compound which enables the cell damage. See, in cell itself, there are some reducing agents available. Cytosine amino acid, glutathione, right? A peptide containing cysteine amino acid. So these are available, reducing agents are available to reduce this copper 2 to copper 1 under physiological conditions. Right? Now, you see this, these compounds, they don't need your cytosine. Sorry, they don't need your cysteine amino acid. They don't need glutathione. They can self-activate the DNA damage and then bring about the DNA cleavage. So we have shown very recently that these two mixed ligand compounds of copper are remarkable reagents in displaying more prominent cytotoxicity than cisplatin. Obviously, it is due to the hydrophobicity accorded by these ligands on the copper. So Once you are able to demonstrate that these compounds can bind to DNA much better, then you have a way of explaining, illustrating the cytotoxicity of the compound. So this Mithu Sarma from Tejpur University, Central University of Tejpur in Assam, which where I am visiting often. So Mithu Sarma made novel compounds of this nalidic acid. Okay, she isolated fixed ligand compound with ethylene diamine. See, this is nalidic acid. This nalidic acid, another nalidic acid, right? And when we have this nalidic acid, they can bind like this. But this is insoluble in water, so we cannot investigate. But when we introduce ethylene diamine, see, on one side, like we showed already in our previous paper, this nalidic acid by producing a water molecule, which brings about this action. So in this compound, what we find is nalidic acid, which binds to copper in a different way as a monoamine ligand. Here is your ethylene diamine, 2 nitrogen. So ethylene diamine hydrogen bond with nalidic. See, when you have a different co-ligand, they bind to nalidic acid differently. So depending on the co-ligands, we can devise newer types of metal complexes. So these compounds can be acting as probably better DNA cleaving agents. So we generated, devised and generated better cleaving agents, copper cleaving agents, biocompatible molecules. See, we have an EPR spectrum, U visible spectrum. This is EPR spectrum of the compounds. Is a very good textbook EPR spectrum for these complexes, right? This shows that the nitrogens are all bound to copper. So we did the titration, interacted the complexes with DNA, and then showed that they actually bind to DNA. So binding studies we did like this. By just simple Jobs variation method, we found that 2 moles of our molecule bind to DNA. We investigated their binding constant. We also docked them on it's called docking on this DNA synthetic, computational DNA, and we showed that these molecules are bound more strongly than this one. See, we got some values which indicate that the compounds are able to bring out, distort the DNA. It's a very big molecule, see. It binds to DNA and brings about conformational changes to the DNA and make it susceptible to cleavage, right? That is how such molecules act as anti-cancer agents. We have done this type of study, document study. See, these molecules bring about a lot of distortion on the DNA. This copper ethylene diamine nalidic acid. Whereas ethylene diamine does not bring about a binding, distortion of the DNA. This is able to bring, a lot of distortions, and we have used this computational method and see they also cleave DNA very well. Not very much, cleave DNA very poor cleavage. So the IC50 values are also very. I think we're able to come. Though we're able to design very good molecules, they are not good cleaving agents. They are not good anti-cancer agents. That's what we found from cytotoxicities. We needed more than 200 micromolar copper compound to design, right? So I'm able to now tell you how the molecules which we have designed, that is mixed ligand compounds of copper. On one side we have phenanthroline, which can recognize DNA when the ring surface is increased, right? They can be better binding, better reagents, better chelating agents, better recognition elements, DNA recognition elements. When you change these ligands, see, you have large changes in the inorganic chemistry of copper, reduction, etc., spectroscopy, EPR, everything. So, depending on this co-ligand here, amino acids, everything, they get involved in DNA damage. They bring about steady crowding around this copper, and the coordinate what I said can bring about the cleavage. So these are the principles we have brought about throughout our work, and we continue to do this type of studies with the help of a fellowship from the Indian National Science Academy, that is senior INSA senior scientist. So we are able to do continue to do much better work. We have devised ligands which can self-activate this cleavage, self-activate DNA damage, and then bring about apoptosis. So thank you for your patience.